Great, thank you. And I'd like to start by saying thank you to ICCOS for the support for this project. So we're going to talk about really more metabolic engineering today, metabolic engineering plans for increased biomass production. And I'd like to start by introduce myself. I'm Ryan Singer. I'm collaborating with Dr. Linda Gillespie from biochemistry. She's joined us here at the back. We also have one graduate student, John Yen, who's here as well. So the research goal, so we would like to produce a computational tool that's going to simplify metabolic engineering for us. In general, we would like to be able to identify metabolic engineering targets. So these would be genes for overexpression or gene knockdown. So again, this is our research team, and we have the following objectives. First is to obtain a working genome-scale metabolic flux model of Arabidopsis, and I'm going to talk about exactly what that is here in a few slides. We're going to derive metabolic engineering strategies using a new approach that we call flux ratios. And from that, we're going to identify metabolic engineering targets that actually produce these plants in the Gillespie lab and test them for cellulose accumulation. And finally, we're going to take the results, our experimental results, and feed back to the modeling process in order to have an iterative development and improvement of the modeling strategy. So, as we start, we have to think about motivation. Why do we want to do this? Really, we're talking here about time, and we're talking about laboratory resources. And obviously, when you're talking about plant engineering, you're talking about something that's going to take a long time. This is also the case with bacteria. And we want to minimize the amount of time it takes to engineer a new plant, and also minimize the amount of laboratory resources that go into that. So the approach, what we'd like to do, so we talk about how you do metabolic engineering. You can do this by overexpressing genes, you can do this by knocking out genes, or even knocking down expression of certain genes. We would like to start by doing this on the computer, see what type of phenotype results, and then from those results, be able to selectively choose what we actually go and work on in the lab. And so next, we use genome-scale metabolic flux models to do this. Now I want to explain exactly what this is. So I say here, simulate global metabolism. So we're talking about models of organisms that represent, say, every chemical reaction and every metabolite in the cell. So these types of models have been around for about 12 years or so. And really what they do is tie growth of an organism and product formation to the substrate uptake rates. So we can see how, or simulate how substrate enters the cell, how that substrate gets broken down, how macromolecules are formed, and how energy and reducing equivalents are formed, and finally how that is put back together into growth and the formation of products and byproducts. So hundreds of these models now exist. They exist mainly for medical applications, and relatively few are related to biotechnology, and I'll tell you even fewer are related to plants. So what these models are, we break down metabolism into a system of linear equations, and I'm not going to go into the details of the mathematics here, but what I want you to know is that we end up forming a stoichiometric coefficient matrix, and in this matrix, each reaction inside the cell is represented in a column, and each metabolite in the cell is represented in a row. And these numbers that you see here are the stoichiometric coefficients of a certain metabolite in a certain reaction. And if it is a positive number, that metabolite is being produced. If it's a negative number, that metabolite is being consumed. We also have V, which is a vector of flux values. And there is a flux value for every single reaction inside the cell. And this is actually what we're trying to solve for. If we know all of the fluxes going through all reactions, then we have a global picture of metabolism. So with these models, commonly we have the number of reactions are greater than the number of metabolites, and so we use linear programming with an objective function. And this process is called flux balance analysis, or FBA. Now, commonly with FBA, this is mostly applied to bacteria, but what we have is when you're using linear programming, you have to identify an objective function, and most people say they want to maximize cell growth rate. And this is an example of a simplified metabolic network over here. And when that happens, if you are looking to produce a certain product, well, your product may not be produced computationally if you are maximizing growth rate. You may only be producing those chemicals that are required for the cell to grow. Otherwise, you can change your objective function, and in this case, produce your synthetic product. But when this happens, you may sacrifice growth heavily, even go to no growth. So the compromise is to actually maximize the rate of product synthesis while also producing everything that is needed for the cell to grow. And so people have pursued this strategy, but what is found is that it takes a whole lot of metabolic engineering to get this to happen. So you have to overexpress and downregulate several enzymes. This can be even in the hundreds. And so this is a very complicated thing to do. So we wanted to ask the question, what can we do experimentally? What can we implement in the lab and do fairly easily? Well, we know it's easy to knock out a gene. You can simply constrain the bounds of a reaction to zero. What's hard to do is to overexpress a gene or knock down a gene. So simply manipulating the bounds of the flux through a reaction we found is not correct. So part of this is theoretical development, which is going to enable us to simulate overexpression and knockdown. The concept, in short here, is that when we have a particular chemical or metabolite in the cell, oftentimes multiple enzymes compete for that limited metabolite pool. So in this case, acetyl-CoA, a very central chemical in central carbon metabolism. It can be used by a lot of different enzymes, go through a lot of different reactions. So what we want to do is maximize the conversion of acetyl-CoA to our particular product through this enzyme. So the way that we can do this mathematically now is start to define how these pathways are used. For example, and we can set this up mathematically by a flux ratio. So we can consider the flux going to our product relative to all of the possibilities that where acetyl-CoA can be considered. And in this case, the example I've shown is that we can metabolically engineer by either overexpressing this enzyme or knocking down these enzymes, these competing enzymes, to where the flux of acetyl-CoA, 90% of it is going to our product. And we can represent that mathematically. We can actually rearrange this equation into a form that can be inserted into our stoichiometric matrix directly. Now, once that is done, it becomes easy for us, and these are some of the details of how this is done, but now we can start to manipulate this ratio in silico and start to calculate a whole wide range of new phenotypes. And we can start to screen those to pick out the one we want. And it may be that your phenotype gets produced when, say, 65% of the acetyl-CoA goes to your product. So we now have the capability to create what I call fine-tuned metabolic engineering strategies. And we put this together into a case study where we did simulations on Arabidopsis, and we've also done simulations on bacteria and yeast and so on for validating and improving metabolic engineering strategies. And here is, this is John. So we took a look at Arabidopsis, and this is our simulation study. We see that this is the pathway leading to the production of cellulose. And there was a group, overexpressed UDP, glucose pyrophosphorylase, I believe that is, overexpressed this one heavily. They found they increased cellulose by 10%. So given this, this, we're able to consider the flux ratios around the UTP metabolite. And I was able to ask John then, okay, if they increased it by 10%, what is the actual upper limit of cellulose overproduction by Arabidopsis? Can we calculate that? And it turns out that we can. And in order to do it, we have to first start with a genome scale model of Arabidopsis. And so John went out and he found the Aragem. It's an existing model of Arabidopsis central carbon metabolism. It counts for over 1,400 open reading frames. I realize that it's small for a plant, but it's fairly large for central carbon metabolism. And so John did some simulations, and I won't go through the details of this, but the result is actually Arabidopsis can increase cellulose content by 30 to 50% without losing its growth rate. And so that, I think, is a very interesting result. And so then the next step was to say, okay, what other targets can we find? Okay, this group already found this target. What other targets can we find, and can we produce those in the lab? So we implemented this flux ratio scheme on a genome-wide basis. And so we started analyzing this for several different targets, and this is what John came up with. He found several enzymes where you reduce function of a particular gene, you should get increases in cellulose, and several where you overexpress a gene, you should also get increased cellulose production. And the ones I have bolded here are the ones that we decided to look at in the laboratory. So that's what I'm going to tell you about next, are the experimental results of what we've seen so far. And I'm going to do my best to explain this. I'm not a plant biologist or biochemist, but I've been coached very well by Dr. Gillespie and John in the back there. So where we started, we decided to overexpress the mitochondrial malate dehydrogenase and chloroplast malate dehydrogenase, and we obtained an expression vector from Dr. Pelot and PPWS. We also looked to knock down or knock out allo-lactate synthase or ALS gene, and we obtained a tRNA insertion mutant. This was a heterozygous mutant from a collaborator, but also this is from actually a Arabidopsis research center. And so some of the results, the first one that I'd like to show is that the mitochondrial malate dehydrogenase overexpression actually did get us bigger plants, and we're currently testing those now for cellulose content. And this doesn't show up too well, but what I want you to take away from this slide is that when we overexpress the mitochondrial malate dehydrogenase, it does indeed end up in the mitochondria. It doesn't go anywhere else. And they've obtained much better pictures than this that are going to be used in publication later on. Some of the results that we've obtained from the chloroplastic malate dehydrogenase overexpression leads to something called variegation. I admit this is a term that I hadn't heard before I saw these plants. But as you can see, these have a wildly different phenotype. So if this is a normal plant, you can see that you see a discoloration of this plant. Now, what we've decided is that this plant doesn't look to have increased cellulose, but there's something very interesting going on with these plants, and it's very interesting from plant physiology and biochemistry perspective. And what we're doing is trying to understand how overexpression of the chloroplastic malate dehydrogenase affects reducing equivalence in the chloroplast and actually leads to a wildly different phenotype. So what you can take away from this is that we've developed now a strain that can be used to actually better study the chemistry of chloroplast, which is very of significant interest to renewable energy. Some of the interesting results we've seen with this strain, variegation is recovered under high light. So I asked the question, why does this happen? I'm not sure we understand yet, but plants are sort of white and green under low light, but when they expose them to high light, they're all green. Some of the things you can take away from this is that in the parts of the leaves that are green, we see very high concentrations of chloroplast in the bleached leaves. We see very low concentrations. You can see also that we have very defined cellular structure in the green leaves, and is actually missing here in the bleached leaves. So this is something else related to physiology that will be of interest to study actually why this happens. We've also done protein analysis on these particular plants and have noticed that the chloroplast malate dehydrogenase is actually absent from these bleached part of the leaves and is overexpressed in this part. We have also started to collaborate with Ava Chalakova from PPWS to do metabolomics on different parts or the different sections of these leaves. And some of the things that we have found is that they are vastly different in their metabolite levels. So, and one aspect of that is it's highly enriched here in ornithine, which is a cell stress signaling molecule. And so what we see is some very, very interesting biochemistry going on here. So, and actually I was asked by John not to present this, but so these are very, very preliminary results related to the owl-acted ALS knockdown strain that we obtained. One of the things I should say about this, we are in the process now of obtaining, we've come to figure out that we can't obtain a homozygous knockout of the strain. This gene is lethal if you knock it out, so now we are trying to knock it down by chemical methods. This result that was obtained was very preliminary early on, and I will also say it's from a heterozygous strain. So we have much more to come with this particular strain, but I just wanted you to know that work is in progress here. From that, we have the following highlights. We've submitted one publication from this. Funded projects, none yet, but we have a lot of very good preliminary data that we're very interested in communicating and trying to propose some new research. With that, I'll be happy to take any questions. Yes?
Why is that your wild type doesn't express NDH?
So this one here? That's actually a very good question. You know what I will do? This is well, we have the answer right back here. Why does the wild type not have? Why is MDH not showing up in the wild type?
It was just the transgenic protein. It was
just for the transgenic.
No, the wild type doesn't have the transgenic form of the protein. We're just simply detecting the transgenic version, not the native version.
We're putting out a tag.
Yes,
native
version.
Correct.
Very good question. I wondered that myself when I was presenting this. Yes.
So the dot product S dot V equal to 0. So what are you conserving? I mean, that normally means the dot product equal to 0. You're conserving some quantity, right?
Yeah, that's right. So S dot V is equal to 0. You conserve. Well, I would say you're conserving mass, but you have here in all of your reactions, we have reactions that are transporting also in and out of the cell, and also in and out of the extracellular environment. So that's how ultimately that's how the whole thing works.
So the mass conservation.
That's
ultimately,
exactly right.
Any further questions? Okay, let's thank our speakers. And that wraps us up.
Okay, well, thank you all for staying through a really great series of presentations today. Appreciate the efforts that all of the folks have put into preparing these talks for the reviews, and look forward to great things coming from this. Thanks. All right, that's it. Yep, good one.
Good job, man.
Oh, yeah. Appreciate it. Very good. Oh, yeah. No, it was really impressive to me.
Yeah,
I know what that's all about. So this is our proposal for IoT. Oh, I got it. Yeah.
well, guys, I'm convinced that we have not seen confusing. Sounds discuss
We're facilitating.
That's why I spit on it.
Most of our work is actually in bacteria.
That's one of the they just call them ice cream. And someone said they should have global. I was thinking of coming up with IS. Coming with IS.
It does. You know,
but this is
model of cancer cells.
how do we take sides giving you program. Okay, so that's and show the right
Let me show it's better. I know, I have a new first now. Just started today.
Okay, just one year.
Yes, you can try to I can do that. Okay, just Nano energetics. I'll definitely do that class.
That's what I'm saying.
Yeah, but I mean, let's hear that.
I want to redistribute pieces where they belong. I don't like odd numbers, so it's nice to meet you. Okay, I got yours. Great job, students.
Jeff is very excited. Jeff finally went to the doctor today.
There's so much of good stuff. No, it's for us to have. How good is this? How do we match against it? You know, we are sitting here.
pull around what it was because there was something in the bio that I think it sounded right.
We need that something.
Okay, well, let me send that. I'll look. Okay. I got to teach a bunch.